So we need to look at the evidence for evolution, and a lot of this evidence Darwin knew, some of it he didn't. So we'll look at the evidence now for evolution. There are different lines of evidence, and we'll go through each of these. So the evidence could be in the fossil record, could be in biogeography, and that's just studying where living, so bioorganisms, are located based on geography or land masses around the globe. Comparative anatomy and embryology, molecular biology, and also in lab and field experiments. Those are the different lines of evidence that can all support evolution. So first of all, the fossil record. One thing on the fossil record is it's incomplete. It will always be incomplete because you need very unique environments to fossilize organisms. Most organisms that died did not leave fossils behind. They were exposed to wind and rain and temperature and predators that broke them all to pieces, and they just degraded. So very few organisms are actually fossilized. Some environments that commonly fossilize organisms are tree resin, but you have to be a certain size, so pretty small, to be fossilized there. Tar pits, well, you have to live near a tar pit to fall into one of them as you die to be fossilized or preserved there. And lake bottoms or kind of stream beds at the curves of the streams where the water would cover you up and then not be disturbed. And not all organisms would die at a lake or at a bend in the river where it wouldn't be disturbed again. So you need a really unique environment to make fossils. A fossil, really the soft parts decay and degrade away, and the hard parts, the calcium, is gradually replaced over time by minerals, and it turns into rock. So fossils are actually rock, and there are some organisms that we have really good fossil records for. Horses, for example, actually evolved in the Americas and then migrated and were later brought back from Europe by humans moving around the globe. But we have lots of fossils showing lots of extinct species of horses and how they gradually changed into modern horses. We can also date fossils using different radioactive isotopes and how long it takes them to degrade over time. And there are several isotopes that span different time ranges, so we can even compare different readings to make sure they're consistent, so we can know how long ago something died. And then we have things like Tiktaalik. Tiktaalik is a fossilized fish that in its forearm, its front leg, it had all the same bones that you and I do in our front leg. So kind of the start of a wrist and fingers to form the front arm. That's the first time we see that in the fossil record, is in this fish. Biogeography is the next line of evidence, and this just shows kind of the distribution of species, and that many species resemble those in nearby environments. So if you remember the Galapagos Islands, very nearby was the coast of South America. The species on the Galapagos resemble or look like the ones from South America, only the environments are completely different environments. This shows that they're probably not designed from scratch to fill a niche. They're moved there, they migrate or are blown accidentally, and then they adapt to their local environment. Some examples of that are looking at marsupial species in places like Australia and comparing them to mainland species. So the sugar glider looks pretty much just like the flying squirrel, only the sugar glider is a marsupial. It has a pouch to grow its offspring. We have this numbat, which looks very much like a giant anteater. Again, one's a marsupial and one's a mammal. The Tasmanian wolf, which is, by the way, extinct, and the gray wolf. Again, they look very similar, they behave in similar ways, have the same type of ecological niche or habitat, except one's a marsupial and one's a mammal. Another one would be the honey creepers of the Hawaiian Islands. This is a bird related to the mainland finch, and perhaps one or two or several honey creepers were blown through a storm or accident onto the Hawaiian Islands, and now they've adapted. So the original ancestral honey creeper looks like this, and now in Hawaii we have this wide variety of beak shapes in the honey creeper, and they're all adapted to different food sources. Looking at comparative anatomy and embryology, so we have vertebrate embryos that we can compare, and really it's the embryo that's impacted first by genetic changes. When you compare embryos of, here's a shark, a turtle, a chicken, and a human, these embryos all have the same parts, and then they start to show differences as they develop. It's sort of like you have an instruction manual for living organisms, and all animals start with the same set of instructions and then add or delete information as you move along. We have homologous structures, and homologous structures are body parts in different organisms that are similar, come from the same ancestor, but are used in different ways. So we have a common ancestor, but the descendants were exposed to different environments and needed to find different solutions in order to survive, so it diverged and became different. An example of that would be found in your arm. If you compare that to the front arm of lots of other animals, or front leg, you see the same bones. So you have the humerus, radius, and ulna, your wrist bones, and your finger bones. Those same parts are found in a horse. Only difference is they still have the humerus, it's slightly shorter and stockier. The radius and ulna during development fuse together to form one very strong bone. They have all the same wrist bones, they're slightly different shape, and they have the finger bones. Horses actually walk around on their basically fingernails and toenails. All the same bones are found in a bat, except they're elongated and slender. So it still has the humerus, radius, and ulna, and the wing is composed of very long fingers that are webbed, and that web connects back to the body. A porpoise, this looks like a dolphin, still has the same bones, even individual finger bones, but it's all encased in the fin. So that shows homologous structures. Convergent evolution is kind of the opposite. Convergent evolution starts at two different points, and then these organisms are faced with kind of the same challenges, and they find the same solution. And that's convergent evolution. This is true of insects and bats. So they start at completely different points, a mammal and an insect, and they both found that flight helps them to survive, so having wings. There's also something called vestigial structures, and vestigial structures are basically leftover structures. An example of that would be in this little species of vampire bat. The vampire bat uses its teeth, and this is a chicken leg it looks like, bites a chicken, bleeds, and it licks up the blood, and that's how it survives. At the back of its mouth, though, it has molars. They're not needed, it only uses this front set of teeth to bite into the flesh to make it bleed, but these molars were once used in an ancestral species to chew food like fruit and insects, but they're no longer used. They're just leftover organs. Another example of vestigial structure would be the human appendix. In evolutionary history, humans, our diet would have been very similar to that of chimpanzees and gorillas. And if you've ever watched nature films, they eat a lot of things that look unappetizing. So lots of things that look like sticks and leaves and bark. We simply couldn't survive on that diet anymore because our appendix doesn't function the way it used to. Their appendix still houses bacteria that will help break down the cellulose in those food items. So basically it ferments the food for them or helps break down those things to give them nutrients. Our appendix doesn't house that bacteria anymore because as our diets changed, it selected for smaller and smaller appendixes. Some people don't even have an appendix anymore because it's been selected out of their genes. So humans also have vestigial structures, and there's lots of vestigial structures. So whales have remnants of a pelvis and sometimes even leg bones in kind of the lower half of their body, but they don't need the legs anymore, but they evolved from a four-legged animal on land. So that's a vestigial structure, kind of leftover leg bones in some whales. Embryology also looks at the embryo and development. So which of these is the human? So you've got A, B, and C, and they all have similar parts. They all have gill slits early on development. They all have a tail. So which one of these three would you guess to be the human? Now, if we were in class, we would take a poll, but just think of which one of these you believe to be the human. And these are actual photographs, not doctored in any way, just photographs of embryos at the same stage of development. And this one is actually the human. Here's a bird, and here's a reptile. But they all look very similar. They all have the same parts. In fact, the human looks the strangest of them all in this particular photo. Another line of evidence is molecular biology, and this is looking at the genetic code of DNA and also the structure of proteins that result from DNA. And if you compare humans to other species and look at, in this case, we're looking at hemoglobin, it's part of your blood. There's 146 amino acids in hemoglobin. If you compare those to a rhesus monkey, they have about the same number of amino acids, but 8 of them are different. And humans and rhesus monkeys are fairly similar, but it's calculated there's about 23 million years of evolution between humans and rhesus monkeys. If you compare this amino acid from a human to a dog, now there are 32 changes. There's about 46 million years of evolution between a human and a dog. As you get further and further away on an evolutionary time scale, you have more and more differences in hemoglobin. And this is true across the genome. So any section of DNA and any protein that you could pick out to compare, most of them will show this type of pattern. That as you get further and further away on an evolutionary time scale, there are more and more differences. And that's amazing to think about. An idea proposed 150 years ago, before we even knew that DNA was a genetic material, before we even knew that chromosomes carry genetic information, back in the time where we still thought gemmules were in charge of all of this, Darwin predicted natural selection, and the molecular mechanism that causes that is DNA, and cutting-edge modern molecular biology is supporting that idea that's over 150 years old. So very, very cool. Also, lots of lab and field experiments. We've already talked about the fruit flies at the beginning of this chapter. Golf course grass is another example of a large-scale experiment. Golf courses are planted with the same grass across the entire course. But what happens is different areas of the course are mowed at different times. So on the putting green, it's mowed constantly because you want to keep that nice and clipped and short. On the fairway, it's mowed down sometimes. And in the rough, you let it grow because it's supposed to be just long grass. What happens gradually over time, based on these different mowing schedules, is the plant adapts. The ones that are on the putting green, they'll mature much faster, produce seeds right away very quickly. The ones in the fairway, they'll mature slightly slower, and they'll produce seeds more slowly as well. In the rough, they have plenty of time to grow, so you get these nice, big, long grasses that gradually mature and produce a few seeds. So they adapt or change based on the mowing schedule. This is also shown with bacteria, unfortunately. In the 1940s, pretty much all bacteria was susceptible to penicillin. So you could put Staphylococcus, which is a common skin infection or skin bacteria, on a plate, put a little disc of penicillin in the middle, and you have this very wide kill zone where this penicillin diffusing out kills off all the Staphylococcus. Well, over time, that kill zone got smaller and smaller until today you can put penicillin at the middle of a staphylococcus plate and it will grow almost right onto the disc. If you ever get to take microbiology, you'll do an activity like this and you'll use a variety of bacteria and lots of different types of antibiotics, and you'll put the little disc on there and look for the kill zone. And as an instructor, this is always a very frustrating lab because some bacteria is so resistant that there's no kill zone. So students always struggle because they simply can't find anything to measure for the kill zone because it's so resistant it grows right up onto the antibiotic disc, which is a huge medical problem. So, thinking of those lines of discussion, or lines of evidence, you need to prepare for your evolution discussion. Your assignment is to bring in 2 examples, so at least 2, and you can pick any 2 of these. So you can pick to do the fossil record and molecular biology, or biogeography and lab experiments, or comparative anatomy and biogeography. Whichever 2 you want, pick 2 of these lines of evidence and research them. And online in Blackboard, it gives a description of what's acceptable research, but if you're using online sources, you need to have it from something that's not a .com. So .com, anybody can say anything. It must be a source from a .org, .gov, or .edu, because those sites have to support what they say. If a .gov, .org, or .edu says something false, other people can call them on it and they'll have to change it. If I say something false on a .com site, I can say whatever I want and nobody's ever going to make me change what I say there. So don't use any .com sites. You have to use actual research information that has been peer reviewed, and that's most likely to be found on a .gov, .org, or .edu site. Or you can use magazines, things like that as well. So National Geographic, Scientific American, those types of things that would also publish information that's peer reviewed. So find 2 articles that give lines of evidence. And what you'll need to do in class, you don't have to write anything about the articles, but read through them and make sure you're comfortable being able to tell the class about them, because we'll go around the room and each individual in the class will give a brief summary of what that article says, what that line of evidence says. Try to use examples that I have not gone over in this lecture and that are not in the book to get full credit for this. They have to be kind of your own examples that you found. Also, the same day we do the articles, after we go around and talk about the articles, we're going to do a discussion activity. So you need to organize your thoughts for this discussion activity. You need to type up, it must be typed, a paragraph or 2 explaining your answers to this question. So look at the controversy surrounding evolution and religion, and based on your background, what you learned in class, and what you learned from your articles, explain why the theory of evolution, in your opinion, does or does not challenge spiritual beliefs. And there are no right answers and no wrong answers for this. You just need to type up what your personal ideas on this are, and keep it respectful. So you'll only lose points on this one if you don't do it at all, don't address the actual question, or if you're disrespectful. So keep it respectful and tell me what you think of this question. And have that prepared, because we'll cover this in class as well in small groups. Last thing that you need to do for this chapter is follow the link, and again it's available on Blackboard in our lectures section. Just scroll down and watch the video, What Darwin Never Knew. So remember, Darwin did his research 150 years ago, so he didn't know a lot of the information that we know today. So the video, What Darwin Never Knew, gives you a better background as well.